Welcome back, everyone. It is now time for CombiGene to present and CEO Jan Nilsson. A warm welcome, Jan. Please go ahead.
Thank you so much. Thank you for the invitation. So let's see if I can see my slides. Yes. As the moderator said, my name is Jan Nilsson. I'm the CEO of CombiGene. We are a gene therapy company. We have two projects based on AAV viruses. One is CG01, the Epilepsy project, and CGT2, which is a Lipodystrophy project. And in CombiGene, we have gathered a lot of expertise over the last 5 years when it comes to developing viral vectors. For example, in the vectors themselves, preclinical studies, including biodistribution and tox studies, development of GMP, classified manufacturing methods, and the scale-up of those methods, which is very important in our field. And of course, strategic regulatory work and also in clinical studies. Gene therapy is a very exciting field within pharmaceutical development. It has revolutionized our way of treating diseases that we were not able to treat at all 10 years ago, for example. We can replace defective or missing genes. We can increase the expression of already existing genes. We can actually cure diseases rather than just address the symptoms of the disease. We have a unique opportunity to target our treatment. This is precision medicine, exactly. So we can have a high degree of efficacy in the target area and very little outside that. And over the last couple of years, we have seen a number of products that has been approved based on gene therapy in both the US and Europe. And apart from the CNS area where we are active, the major areas of interest are in oncology and cardiovascular disease. Our business model is to develop effective gene therapies for severe life-altering diseases where there currently is no adequate treatment available. And we source our assets from external networks, and we develop them from the lab bench to the first in human study. And if you look at the lower part of this slide, you can see that for drug candidates, which have a broad indication, we will seek partnerships for the continued development and commercialization after the first in human study. Whilst when it comes to a narrow indication, a niche indication, we can either do that, the same procedure, or we can go all the way ourselves to the market. We have, as I mentioned, two projects, CG01, which is in late preclinical stage, targeting severe epilepsy, and CGT2, which is a molecule for lipodystrophy, which is in early preclinical phase. And as you can see at the lower part of this slide, we have received grants from Horizon 2020 of 3.36 million euros for our epilepsy project. And for CGT2, we received earlier this year almost 900,000 euros for the development. This is a fantastic validation of both the science, but also the team behind it. Sorry. So a few words about epilepsy. It's a common disease, and that in itself sets up the sign for most other companies working in gene therapy. If we look at the US, the 5 big countries in Europe and Japan, we have almost 5.7 million patients that carry the diagnosis epilepsy. 1/3 of those patients are drug resistant, i.e. the current available pharmaceuticals will not alleviate them from seizures. And having seizures means that you have a severe, significant impact on your quality of life. You cannot take certain types of jobs. You cannot ride a bike, drive a car, swim in deep waters and whatnot. And on the right-hand side of the slide, you can see the different types of seizures that an epilepsy patient can have. And the ones we are interested in are the ones with focal seizures. So if you add then focal seizures and the pharmacoresistant patient, these are the ones that we target, that constitute more than 1 million people in this part of the world that fit that description. Now, going into the mechanism of action of CGO1, which as I mentioned, is an adeno-associated virus, a small virus which doesn't cause disease in human beings, but it carries the genes then for neuropeptide Y and Y2, which are two naturally occurring substances that we all have in our brain. And if you look at this cartoon, then this depicts the interaction between two nerve cells. And when nerve cells signal, they use neurotransmitters. And the common denominator for an epileptic brain is that you have too much of a substance called glutamate, which makes it too easy, if you like, for the cells to signal between themselves. And when the signaling increases, and if it becomes synchronized, then you can develop an epileptic seizure. Now, here we have our patient who have a focal pharmacoresistant epilepsy, and the focus here is in the yellow part, which you can see deep in the brain. We can then go in with a catheter from the back of the skull and inject our AAV virus, drug candidate, which then will infect the neurons in that volume of the brain. And then the neurons have an increased capacity to produce neuropeptide Y and to produce more of its receptor, the Y2 type. Neuropeptide Y is normally stored inside the cell, but when the signaling increases, the cell will release neuropeptide Y, it will bind to the receptor, and as a consequence of that binding, the cells will normalize the glutamate release, hence you will not develop a seizure. So I hope you agree with me when I say the gene therapy is a revolutionary treatment option for these patients. It is a first-in-class disease-modifying therapy. We actually address the root cause of the disease rather than just treating symptoms. We have a very long-lasting effect. That is a feature of gene therapy. If we look at our colleagues that have used the same type of virus but treat Parkinson's disease, we know that the patient express the proteins of more than 10 years after the injection, after one single injection. And in monkeys, the corresponding time span is actually 15 years. So it is a long-lasting effect. It's safe and effective. We have so far not seen any signs of side effects in our animal studies. And we are utilizing the body's own mechanism to handle a seizure. And here you see a cartoon of a drug candidate. So we have proof of concept of CGO1. On the right-hand side here, you see an arrow. On the upper part of that arrow is the development of the human disease, epilepsy, all the way from a precipitating event all the way through to eventually surgery, which is very uncommon actually. On the lower part of the arrow, you have the corresponding rat model that we have used. These rats have spontaneous recurrent seizures. They will get either our drug candidate or they will get an empty virus. And when we compare then those two groups, we can see that the animals that got our drug candidate have significantly fewer seizures. The seizures are significantly shorter, and we see animals that became totally seizure-free. We also look for side effects in this model, and we couldn't confirm any side effects so far. So in conclusion, we have proof of concept of CGO1 established in a chronic rat model. Now, what about the market potential for this? I already alluded to the upper two rows on this slide previously when I spoke about epilepsy. It's a very common disease. If you go to the third row, it says 47,000, and that is the number of drug-resistant patients with a focal epilepsy that are diagnosed every single year. So these are new patients. And if you just focus on that group, and we could target maybe 10 to 20% of those as in terms of market penetration, that would translate into what you can see for 4,700 to 9,400 patients per annum. And if the treatment cost is somewhere between 134 and 200,000 US, that will give us a market potential between 750 million to 1.5 billion dollars a year, without touching the already existing patients. I'm sorry. Then we turn our attention to lipodystrophy. This is a rare disorder, and it's characterized by that the patient has not a normal fat distribution in the body. Quite often they lack subcutaneous fat altogether, so the body then stores fat in other locations, like the liver, which is a problem. There are approximately somewhere between 1,000 to 2,000 patients with this diagnosis in Europe and US. And it's a severe disease, it's a rare disease. We think that we have a possibility to apply for and to get actually an orphan drug designation once we have enough data. And currently there are no approved treatments for partial lipodystrophy, the group that we are targeting. And the aim of this project is to break that vicious circle of liver lipid accumulation and to normalize liver lipids. Because if you have a lot of lipids in your liver, these patients will develop very high blood levels of lipids. They will develop high blood pressure, and they will also have develop a type 2 diabetes. So they will die very young in cardiovascular disease. We filed for a patent on this project this summer, and right now we are working hard to arrive at a final drug candidate. As I mentioned, we received a grant of almost 900,000 euros for this project earlier this year. Now, and if we are successful in the field of lipodystrophy with this molecule, then we can see other applications which are characterized by a fat liver. And that is for the future to decide. And of course, we have had a lot of synergies and used the experience we have gathered of CG1 when developing CGT2. We have a very experienced team. We have built that over years. Next to me there, you have Karin Hageman, who is my research and development officer. She has experience from both large pharma and smaller biotech companies. And on the right-hand side, you have the project management with Annika, who is an expert in preclinical setting and biodistribution and tox studies. She has actually gene therapy in her PhD. Pernilla next to her have experience also from small and large companies and is targeting the clinical development area. Martin is our CMC expert. He has experience from developing biological products, and CMC is a very important area when it comes to that type of products. And Esbjörn on the right-hand side, he actually was the individual responsible for our proof of concept study, which I discussed previously in rats. So we are very lucky to have him as an employee as well. And then we have, of course, Louise, who is our CFO, who handles all our financial issues. Then if we look at the board, again, we have a very experienced board in this area. Both Bert, Gunilla, Jonas, and Per have experience from companies like ours, so they know very much about the challenges and opportunities which we face, and we have a very good interaction with the board. Peter, on the other hand, is a financial guy. He works currently as a CFO and has previously worked as a certified public accountant. So he adds a lot of value to the board as well. So what have we achieved so far? Last year we got a number of preclinical studies. They all yielded positive results. We switched to a suspension-based production method, which is easy to scale, extremely important. First large-scale batch was done last year. This year we got to go ahead from the British and Swedish medical authorities for the remainder of the preclinical program. We have initiated the pivotal preclinical studies with biodistribution and tox, and we are preparing for the first in-human study. And in CGT2, as I mentioned, we are focusing on selecting the final drug candidate. Next year then, we expect to finalize the pivotal studies within biodistribution and tox, and after that we can submit a clinical trial application, and if everything is okay, we can then go on, once approved, with the first in-human study. CDT2, we expect to do a proof of concept study next year. The following year, we continue the enrollment of patients in phase 1/2 study of CG01, and we can apply for an orphan drug application when it comes to CGT2. So I hope I have been able to share some of my enthusiasm for this revolutionizing area within the drug development area. Thank you so much.
Thank you very much, Jan, for a very interesting presentation. Perhaps you could share with us a comment on that you recently announced the initiation of GMP production of CG01 in collaboration with ViralGen. Could you please elaborate on what this means for CombiGene and CG01?
It's very important, I would say. I think I already mentioned that during my presentation, that manufacturing is a key component when it comes to biological products, especially so when it comes to viral vector products. ViralGen in Spain has a suspension-based method, which is used by other companies as well, like AskBio, like Bayer. So we have a state-of-the-art production facility doing our producing our vector. And one should realize that selecting the manufacturer for our drug is like, you know, going into marriage. This is something that will follow the product all the way from now till the end of the commercial period.
And speaking of marriages, you plan to find a partnership for CG01, of course. Could you please elaborate on that process? What can you share with us?
Well, I can share this much. I'm sorry. I have a long experience from the pharmaceutical industry, both big pharma and small biotech companies. And I never had the pleasure of having a product or a project which has attracted so much attention from big pharma. Having said that, we have interacted with several companies, and I don't have any doubt in my mind that we will follow our business model, that once we have more data, we will find a commercial partner for this. We have interacted with several of the companies on the top 10 list in the world.
Mm-hmm. Thank you for that. And to round off the Q&A session, if you would look 1-2 years into the future, what upcoming value creation points would you like to point to from an investor perspective?
I think I had that basically on the second slide from the end, actually. I think this year we will have the initiation and result from the first pivotal biodistribution tox studies. Next year we will do the GMP batch. We will get the release on that one. Good manufacturing practice, that's a batch we need for going into human. We will get the result from the large animals in biodistribution tox. We can do the clinical trial application and henceforth start the first clinical trial next year. And the following year, I expect that we will have the final candidate and do the proof of concept in CDT2. And we can continue to do the enrollment of patients in the phase 1/2 study. And we will do an application for orphan drug designation.
Indeed. No doubt, there's a lot to look forward to. Thank you very much once again for an interesting presentation and for joining us today.
Thank you.